Hello everyone, I hope everyone's having a great day when they start watching this video. What we're going to start talking about today is mitosis. So, sorry to get us switched back to more of a thought process about cells. I know we've been learning about more chemistry, but we're going to go back to learning about cells. I just wanted to make sure that we learned about some of our chemistry stuff because it's really important. But anyways, our mitosis, what we are going to talk about is how cells divide. But our questions for today, as we can get our brains working and started thinking about this, is what happens when we get cut. So just take a second, maybe pause the video. What comes to mind for me is that we first see blood, of course. So especially if it's a deep enough cut, there's a lot of blood. But once it starts to heal, it becomes a scab. And I'm sure everyone's seen a scab, and maybe some people like to pick or tear their scabs. I know every once in a while I would. But underneath our new scab is new skin. It looks a little bit different, but it's still new skin. But where does this skin come from? How does our body make it? It's coming from somewhere, but it's coming from our body. Well, our body makes it through a process called mitosis. We're going to learn all about mitosis coming forward. So by the end of the day, we want to be able to understand the basics of mitosis, and then be able to list and describe the steps in mitosis as well. So let's go. First, we have this video. It's linked in our classroom in the same place as always. So please pause this video and go watch that one. It does a good job of explaining mitosis and just giving a general idea. And it's a short one. It's only 3 minutes long. And so then our basics. So I always like to start with our definition. So our definition of mitosis is when a cell makes a new and identical cell. So for example, if Mr. Max was to undergo mitosis, I would make a new and identical person. So it would be almost like a twin or a clone, and I could get my clone to do all my laundry, maybe clean the house. It would be great. Maybe he would even cook me dinner. So it would be really great if I could easily have mitosis for my whole body, but it only works on a cellular level, on a very small cell-by-cell level. Mitosis is used in almost every type of cell, and it also makes a new cell up to every 24 hours for eukaryotic cells. So this is actually happening quite a bit. It's not something that happens once and we wait a few years. It's constantly going on. And that's how our skin can get repaired, is because we're always making new skin. We're actually losing skin a lot of the time too. Maybe you guys have been sunburnt and your skin peels off, and we have new skin right there. But it is more than just skin. It's every type, almost every type of cell in your body. The first thing we're going to talk about is our life cycle of a cell. So we have a few main things, phases, I should call them. So we have our M phase, our G0 or G0, G1, S, and G2. And our G0, G1, S, and G2 are all summarized as our interphase. We're going to talk a lot about that here in a little bit. And then our M phase stands for mitosis. So M, and it's broken into our prophase, metaphase, anaphase, and telophase. But the idea is, before we even start our mitosis, we go through a growth phase, which is preparing for DNA synthesis, and then a replication phase of our DNA. And then we go through another growth phase in preparation for mitosis. So the idea, before we even start our mitosis process, we have already doubled the amount of DNA we have, because we're building towards trying to make a separate cell. We want to make a new and identical cell. So we start with one thing and we want two, and we need the exact DNA we are starting with in our new cell. So we have to start there. Some important vocab stuff. This picture, I think, does a good job. We're going to be talking about chromatin. Chromatin is kind of this noodle-looking part. It's actually all of our DNA kind of spread out inside of our nucleus. So this is all chromatin. The other stuff we're going to be talking about is a chromosome. So our chromosomes kind of look like these X's. They kind of look like an X. And then the middle of them are called centromeres. Centromeres. The other vocab we're going to be talking about is when we have a one chromosome, once we copy it, they start to become chromatids. So we have one chromatid and one chromosome here. Now we have two sister chromatids because we say sister because it's the same exact DNA. And it is still one chromosome though. And this time we have a bunch of cohesion bonds. And then once it gets kind of separated into more of an X shape, it's still one chromosome and two sister chromatid. And then once our chromosomes get separate, we have two chromosomes, each with one chromatid. So just so you understand maybe some of the words we're going to be using. Just want to go over those. So we have an easy way to remember our stages for our meiosis. We have, I'm not quite sure if I want to say IPMAT-C or IPMAT-C or what yet, but the idea is each letter in this stands for a different stage. So our first stage is interphase, that's the I. Our P is prophase. Our M is metaphase. Our our A is anaphase. Our 5 is telophase. And our 6 is cytokinesis. So those are the stages, and then we just took the beginning of each stage and made a little acronym for it called, I like IPMAT-C, but that can be kind of hard. So IPMAT-C also works, or whatever you guys want to say. So we're going to break down each stage now into a little bit more detail. So our first stage is interphase. So interphase, we have our G0, G1, S, and G2 phase. And this is where the cell spends most of its life. So if a cell replicates every 24 hours, this is at least I would say probably 22 hours of the day. So almost all of its life is in these phases. The important part, this is where our DNA is replicated. And this leaves us with 2 copies of DNA before we even start our mitosis. That's really important. We can see here how we have in this picture, we have our nucleus, our nuclear envelope, and it says chromatin is being duplicated. So we're getting more and more of this kind of messy spaghetti called chromatin. Oh, and then also we have centrosomes, which are going to be important later on, and those are going to start showing up. Then we have prophase. So we have interphase, prophase. Prophase are chromatin shrink to chromosomes. So when we have chromatin in our spaghetti, as they condense and look more like our X, they look more like our chromosomes. And then our nuclear envelope right here starts to break down. We also have these little spindles that start to appear. And this is the longest part, or the longest stage of mitosis. Our next step is our metaphase. So what happened here is our spindle fibers, these little fibers that we had start to show, attached to the center of our chromosomes, and they all lined up in the middle. So when I think metaphase, I think middle metaphase. So we have all our chromosomes lining up in the middle, and they all have a spindle fiber attached to each one. The next part is our anaphase, and this is when our chromosomes are pulled apart. I like to think of this much like if we were fishing and we had a pole getting reeled in. So if you've ever been fishing, you tighten the line as you turn, and that's what happens here, is our centromere down here is tightening this line more and more and more until it can pull apart each one of our chromatids onto either side. So we're trying to separate our chromosomes that were in the middle of our cell. The next stage is telophase. So these are when our chromosomes grow into chromatin. So we were nice and condensed into chromosomes, and now we're going to go back to this big spaghetti mess. Our spindle fibers that are fishing line break down, and then we also form our nuclear envelope. So inside once we have our nuclear envelope, our chromosomes can grow big into our spaghetti mess of chromatin again. And then our last step is cytokinesis. So in this step, we have our cytoplasm split into 2 cells and it divides. This happens a little bit different for plant and animal cells. In our animal cell, we put this what's called a microfilament ring around the edge, and it just kind of squeezes our cell until it's 2 separate. We can see we have 2 nuclear envelopes on either side, so all we need to do is kind of cut it in half. But in our plant cell, which has a cell wall, it's a little bit different. This thing gets made called a cell plate, which is made of our plasma membrane or cell membrane all the way down the middle. And then once that's fully made, then it clips off into the cell wall. So we have our cell wall around it the whole time here on our plant cell, but on our animal cell, since we don't have a plant wall, cell wall I should say, we have a little bit different kind of process. So here are our basic steps. We have interphase. Our important thing to remember is that our DNA is replicated in interphase. We have our prophase, which is our chromatin, our spaghetti kind of mess, shrinks into chromosomes, which look like X's, and our nuclear envelope, which is part of our nucleus, breaks down. Then we have our metaphase, where our chromosomes line up in the middle. Remember, meta, middle. And then we have our anaphase, where our chromosomes are pulled apart from each other. Our telophase is that our chromosomes go back into chromatin, and we form our nuclear envelope. So the telophase is the opposite of prophase. And then our cytokinesis phase, our very last one here, is where our cytoplasm splits into 2 and our cell divides. So now at the end of our cytokinesis, we have 2 separate cells, and each one of those can go and replicate again. So as we continue to replicate, it kind of grows and grows and grows and can expand more and more. So that is our summary. Now that we've kind of worked through it, I found this really cool animation which does a really good job of showing it. We can see right now that this is showing anaphase because we're starting to pull apart our chromosomes right here. So this is also linked in our classroom in the same spot as always, so please go watch this. I think after kind of outlining each one of those, this is a good way to picture what it's looking like. So this is just the start of it. We're going to do some more work with mitosis next week, and then also get into a little bit about meiosis. Thank you guys very much, and if you have any questions, please let me know.